going to introduce the work of my group we have done about 10 years ago. We started 10 years ago, and I want to show you that it takes a lot of time actually to answer this question: can we have climate-proof apples and more generally fruits? And so I am located at University Paris-Saclay with my group, and some people of this group are going to arrive in New York University next year. So first, before starting, I want to thank all of my group. So there is Anthony, who is a molecular biologist, Amandine and Mathieu, who are ecologists. We are bioinformaticians, so Shilong Xio, Aurélie. And there are also a lot of PhD candidates, so Sergio Seila, Yuchi, Maxime, Ronan, and a lot of master students. And without them, I could not show you all the results of today. So how do populations adapt to their environment? This is a key question in the context of global changes, which could include disease outbreak, so you faced it, it's COVID-19, of course, pest outbreak, and also climate changes, which is the topic of today. And so what about fruit trees? So fruit trees are key species in ecosystems. They offer shelter, they offer also food, and in agro-system, of course, they offer food for us. You see here a bunch of examples on species we are eating, actually, but surprisingly, we still don't know a lot of their responses to climate changes. And a very first step to answer this question is to know what is a crop and what is a wild species. And so you need to study the crop and the wild, and their wild relative actually, and you need to understand their evolutionary history. So to that end, you need to go back to Darwin's theory. So since Darwin, we are trying to understand which evolutionary processes are shaping diversity in ecosystem. Why do you have so much morphology, physiology, phenology? And in particular, we are trying to understand which evolutionary processes are shaping divergence. So what I will call divergence is the differentiation among lineages that could lead to new variety, new population, or new species. And historically, we have tried to understand the role of natural selection during this divergence process. So for instance, here between brown bear and white bear, versus the role of gene flow. And this gene flow can intervene through two different ways. First, you can have the establishment of barrier to gene flow, which would lead to the differentiation between the two taxa. Or you can also have introgression of genes, so incorporation of genes into taxa 2, for instance, by taxa 3, which would lead to the divergence of taxa 2 and taxa 1. And so historically, we still don't know the balance between this process of natural selection and gene flow in the divergence process. And one awesome model to answer this question is to look at domestication process. Indeed, domestication is a very recent divergence process during which a crop taxa, for instance here maize, have diverged from a white taxa, here the Thiocynth, the ancestor. And during this divergence process, you have selection, selection for agronomic traits. So here you have the example of Thiocynth versus maize, wild apple versus cultivated apple, white tomatoes versus cultivated tomatoes. It's obvious. And at the same time, you still have a lot of gene flow between the crop and the wild. So what about the divergent history of the cultivated apple? What about the domestication history of the apple? You probably eat apple or have eaten apple once in your life, I bet. And actually, this apple comes from one single species, which is called Malus domestica. This species produces all the diversity of apples you can find on the market. And surprisingly, when we started to study this species, we didn't have a good idea of the role of gene flow and selection during the divergence processes. And when it comes to study the domestication of the cultivated apple, you can focus on four main wild relatives. So first you have Malus sylvestris, which is widespread in Europe here, the European crab apple. Then you have Malus orientalis, which is widespread in the Caucasus. Then you have Malus stiversiae, which is a Central Asian wild apple, which is widespread in Central Asia. And you have Malus baccata, which is widespread in Siberia and Southeastern Asia. And phenotypically, these species have very different fruits. If you see Malus sylvestris, it has very tiny fruit, very sour, like Malus orientalis. If you eat them, The next day, it's kind of complicated, I could say. We have tried in the field. Then you have Malus sieversiae, which has an astonishing diversity of fruits. You go in Central Asia, you can eat, you can survive in the forest. And actually, because of this diversity in the forest, it has been supposed as the ancestor of the cultivated apple, and I will tell you more about that after. And Malus baccata is our outgroup in our study most of the time. He has very tiny fruits. And so today I'm going to show you what are the contributions of this species to the cultivated apple genome, what is the role of gene flow during the divergence. Also, I will talk a bit about the role of selection and the devenir of these wide genetic resources in the context to face climate change. So what about the origin of the cultivated apple? So actually it was Nicolas Vavilov with a remarkable foresight who predicted that the cultivated apple was originating from the Tian mountain. So these mountains are between northwestern China, Kazakhstan, Kyrgyzstan. And if you go there, you can find forests that are the slope of the mountains like that. You can survive in these forests like man versus wild, no problem. There are bears, but that's the only problem. And when he went there, like a long time ago, he said some of the forms in this forest were so good in respect to quality and dimensions that they could be directly grown in a garden. So there were some apple trees that are actually Malus sieversiae that really look like the cultivated apple. And it was the first clue, and later on, his hypothesis was validated with some genetic data when the first draft genome was released in 2010. So we were okay, so the ancestor is sieversiae, but there were additional contributors present along the cycle route that were supposed to have contributed. So scenario being that Malus domestica originated from Malus sieversiae in the Tian mountain, and then it was brought along the cycle route toward Europe. And during that trip, it hybridized with different white species. So different white species could have contributed. First, Malus baccata in Siberia, then Malus orientalis in the Caucasus, and later on, when the Greek and the Roman brought the cultivated apple in Europe 1,500 years ago, you had potentially a contribution of Malus sylvestris. So it was a nice hypothesis, but at that time, we didn't have any genetic clue to validate it. So I just summarized most of the results here. So what we did is that we genotype a big collection of cultivated and wild apple using microsatellite marker. And what we show is that indeed Malus sieversiae is the ancestor of Malus domestica. And then once the cultivated apple was brought in Europe, you have massive introgression by Malus sylvestris into the cultivated apple gene pool. So this was very exciting because we were showing that actually the domestication was more complex than what we thought before, with an initial domestication from one species and another contribution through Y2 crop introgression. And then we went in Iran with Amandine and we sampled a lot of population of Malus orientalis, and we showed that this species also contributed to the cultivated apple genome, and actually there is an additional center of domestication in this region. And later on, when Shillong arrived in the team, we started to investigate the role of Malus baccata. And we showed that this species also contributed to the cultivated apple genome, and specifically to the genome of Chinese variety, Chinese cultivar. So if I summarize, you see it's a very complex history of domestication. You have several contributors and massive Y2 crop introgression. And we show that basically with microsatellite marker and also synonymous SNP. And this is a pattern that we also show in a lot of fruit trees. Like we have shown that as well in apricot, in olive tree, in grape, and of course, you probably know better than me, in date palms. So at that time, we were okay, you have a lot of gene flow from the wild to the crop, but do we have a lot of gene flow from the crop to the wild? And what are the consequences for the wild genetic resources? So we focus on that question in Europe. So we look specifically at the gene flow from Malus domestica, to Malus sylvestris, because we've shown before that it was a major contributor. And we wanted to know if the European wild apple could be threatened by this gene flow, and to answer this question, we needed to quantify the gene flow. We wanted to understand which anthropogenic factors were influencing this gene flow, and whether you have adaptive consequences. So what we did is that we did a big sampling called citizen science sampling code and also field sampling. We also have 300 cultivars that were genotyped with microsatellite marker. And as population genomician, we love to do this kind of bar plot. You probably have seen them, and it's like we love color. And here in this bar plot, you see here the 300 varieties that we have genotype. And so each vertical bar here, you don't see well the red color, but each vertical bar, it's one individual that was genotyped for the microsatellite marker. And with this kind of method, you can estimate the degree of admixture between different gene pools. So you have in yellow the white gene pool, in black the cultivated apple gene pool, and you see here the 300 varieties are mostly black, but you see some cultivars are yellow. And this is the introgression I talked about before, about the contribution of Malus sylvestris into the Malus domestica gene pool. So that was not a news, but about Malus sylvestris, you see that there are a lot of black in the Malus sylvestris gene pool, which means that you have massive crop to white gene flow across Europe, because here you have the average of this introgression rate per country, and you see that globally any country, any population in Europe are introgressed. You see that in Italy, apparently there is no introgression, but actually the sampling was low, and when we increase the sampling, we have seen also introgression. So we were okay, there are a lot of gene flow, but which factor influences gene flow? Is it natural factor, pollinator? Is it human factor? And so what we show is that the higher the apple production and the higher the number of orchards surrounding the natural population, the highest the introgression rates. So clearly we indirectly influence this crop to white gene flow. And then, okay, you can have gene flow from the crop to the wild, but maybe on the long time scale, you don't have any consequences for the wild. So no problem for the wild genetic resources. So you need to know if there is adaptive consequences of this gene flow. And for that, we went in the forest next to Paris, and we sampled some fruits. We took the seed, we grew the seed, we measured their growth rate and also their germination rate, and we also genotyped the seedling to know whether they were hybrid or whether they were pure. And then what we show is that the seedling which were hybrid with the cultivated apple germinate earlier and grew faster compared to the wild apple, which means that potentially this crop to wild gene flow could be a threat to the wild apple on the long term. So at that time we were okay, There are massive gene flow, shaped by us mostly, which have potentially adaptive consequences. So, okay, Malus sylvestris is potentially a threatened species, so we should set a conservation program for this species for future, to face climate changes, maybe. But to settle conservation program, you need to know the genetic diversity of your species. So we investigate the population structure of the European wild apple using genetic marker, and we show that there were 5 main genetic groups, 1 in Scandinavia, 1 in Eastern Europe in red here, 1 in Italy, and 2 in France. And we show using modeling that these groups result from the past climate changes. So maybe you know that 20,000 years ago, there were a decrease of temperature across the world, and most of the temperate species in Europe migrated in so-called glacial refugia, so Spain, Italy, and the Balkans. During that time, the population did not exchange gene flow, so diverged. And then 10,000 years ago, during the Holocene, there were warming across the world, and so most of the species migrated back to the northern part. And so when they migrated back, they hybridized together, and that's what we show using modeling, is that during that migration, you had a lot of gene flow. So this was very interesting for us, because we say, okay, at least we have 5 genetic units to conserve, and we also see that there is substantial gene flow during the intraspecific divergence of a temperate fruit trees. And last but not least, that actually the wild apple responded to past climate changes. So do you have there behind a genetic adaptation, which and how many genes could be involved, or alleles, or maybe epi-alleles? And is there also any plasticity? So with all these questions in our pocket, we say, okay, let's go back in the field, let's sample some seed. So we went on the field with our colleague in Romania, with Gail Vogt, maybe you know her, she's taking care of the germplasm of apple in USDA, and we went in this kind of cool landscape. landscape. It was pretty fun to go in Transylvania and so on. And then we constitute a big collection of seed. And we say, okay, let's settle a conservation orchard. So I put all this logo because it was a scientific adventure, a political adventure, a personal adventure to settle this orchard. I was a genetician initially, not someone doing horticulture. And so we use seed from this 5 genetic group, and we planted them. But before that, we had to grow them, so we sown them just before the COVID. So we had 2,000 seedlings that we genotyped to be sure they were not introgressed by the cultivated apple. And then the lockdown. Awesome, for 1 year we could not check the plant, but then we came back and they were like super high. Some of them were 2 meters tall, so they grew everywhere in the greenhouse. And then we planted the orchard in November 2020 with a lot of people, but it was the 3rd lockdown, so at the end we were just only 20 people. And then we started to sample on the field, so to sequence the leaves and also measure the phenotypes. And so I want to thank all the people who have been involved in this adventure. And this station for us is a way to understand the response of the wild apple to climate change. Not only, we have also a program on pest attack, and especially we want to understand whether you had climate-driven natural selection that led to the divergence of the wild apple, and which genes and how many genes are involved. It's also an ex situ source of science for conservation and breeding program. We have a training and outreach program, and I won't detail it with Amandine. And we have deployed this design across Europe. So then we were, okay, let's have fun and let's plant 1,000 trees. Actually, we have planted enough trees, I bet, if I go back to the talk of yesterday night. And so you have in yellow all the wild apple orchard we have planted. It's more or less the same design as in Paris-Saclay, but smaller. You have 100 trees. And we have also cultivated apple orchard, which are much bigger. And here you have some preliminary result of what we have found on the 2 orchards that we have planted in France and Spain of Malus domestica. And here you have a PCA showing the variation of flowering time and photosynthetic activity. So this is the work of Maxime and Sergio. They are still digging in this data set, and you clearly see a difference of response. And I could tell you more next year, I bet. And as a preliminary step to this in natura design, we started to understand the response of wild and cultivated apple to climate change in controlled conditions. So we use climate simulator. So I don't know if you know this kind of climate simulator. You can precisely simulate temperature, humidity. So we start this adventure with all these people here in 2019. So it was a long time ago. What we did is that we took seed that we sampled from Denmark, from France, from Romania, and from Armenia. And we did a transplant experiment. So we took this seed, we put them in the French climate, in the Romanian climate, Armenian climate, Danish climate. And on each seedling, here, we use an integrative approach. We measure phenotypes, physiology, so photosynthetic activity, carbon-nitrogen reserve, morphology, performance. It was the work of Noemi. And we look also at the gene express, and we sequence the genome of the seedling. And our aim was to test whether you have local adaptation and or plasticity in the wild apple and in the cultivated apple. If you have local adaptation, if you are not so familiar with this concept, it's supposed that you have a genetic basis. So here, if you have local adaptation in our design, we expect to have the fitness, so survival of the seedling, which should be higher, for instance, for the seedling coming from Denmark in the Danish climate, for seedling coming from Romania in the Romanian climate, and so on and so forth. This is the theory. Let's see the data. So here you see the survival rate of the seedling depending on the climate condition. If we focus first on the seedling coming from Denmark and France, what you see is the survival rate of the Danish population is the highest in the Danish condition, climatic condition, and it's the same for the French, it's the highest for in the French condition. So it suggests that you have local adaptation of the French and Danish population. If you look at the Romanian population, it has very low survival rate, whatever the climatic conditions, suggested potentially maladaptation of this population. And then you look at Malus orientalis, for this species, it's like Everything is fine, whatever the condition. And this is the same for Malus domestica. Whatever the climate, it's very stable. So you may have local adaptation, maladaptation. You have all the pattern in Malus domestica and Malus silvestris. And then we were, do you have plasticity? So just if you are not familiar with this concept, so plasticity is the fact that a given genotype can have different phenotypes depending on the environment. And so this is a key trait that could help us face climate change. And I'm not going to detail all these graphs, just to show you that in Malus silvestris, so you have the three populations, so the Romanian in red, the French in green, and the Danish in blue. And we show for each trait their measure. So you have a growth rate, phenotypic activity, carbon-nitrogen ratio, and morphology. What you see is that all the curves are parallel, basically, which means that it's a typical pattern of plasticity. So you have a huge plasticity as well in the apple tree. So once you are settled, then you are plastic to climate. And we're okay, so you have local adaptation, plasticity. If you have local adaptation, which genes could be involved? So we have used RNA sequencing. So we have shown that you have differentially expressed genes, mostly between different climatic conditions, and Ronan is finishing to dig in this data. And Shillong has associated SNP variation with climate variation, and we have found several outliers, but we are still finishing the manuscript, so I can discuss with you about that later. And the aim is still to look whether you have climate-driven selection that led to the divergence of the wild, and which genes and how many genes are involved. So if I try to summarize this part, using an integrative approach, in control condition, we show that there is a huge phenotypic plasticity of the wild apple. And you have also local adaptation for the French and the Danish population. You see the Romanian population is maladapted to its climate. You have a high stability of the cultivated apple to climate. It's really interesting to see that. We are still digging in which genes are involved. We have also this big experiment on the field, because sometimes the results you have in control condition are not the same that you see in this Natura setting. And a big project we are starting, and this is in the context of Maxime PhD, is that we are now predicting the response of different fruit trees to climate change. So we use genomic offset analysis. You combine genetic, you combine climate model, and you can predict to which extent these species can respond to climate change. So Maxime will build the bioinformatic pipeline to apply this analysis to peach, apricot, apple, grape, and olive. As you see, we are diversifying, and we will start on fig trees here, so more diversification. And so I have talked a lot about the process of natural selection and also the role of gene flow during the natural divergence of wild taxa. But what about the anthropogenic selection and its role in the divergence of Malus domestica? So this is what Shillong is finishing his manuscript. This is the year of the manuscript, I bet. He's looking at how many and which genes are under selection during domestication, and especially, as I show you in this talk, you have massive introgression from the wild to the cultivated apple. So where this region are, which genes are concerned, and do we have climate-related genes or alleles that are from the wild population? So what is the response of wild and crop fruit trees to climate change? We have investigated that in apple. And as I told you in the introduction, first, before answering this question, you need to understand the evolutionary history of your species. And what we show is that you have a complex history of domestication of the apple tree with a lot of wild apple which has contributed. You have high level of crop-to-wild introgression that are promoted by us and threatening the species and also potentially genetic resources, drought resistant, but also disease resistant. And we are looking at genes selected during domestication and especially the climate-related genes. So now we have a good idea of the evolutionary history. We have started to launch projects on the response to climate change and especially testing whether you have adaptation and plasticity and which allele are involved. We have shown in control and also natural conditions that both plasticity and adaptation are involved. We are still investigating which genes are involved, and especially this in natural condition. I haven't talked about the project on pest attack, but we have another part of our PhD candidate working on that. And something I haven't started to talk about, but many people talk about also this morning, Etienne, about the p alleles and especially methylation and the role of transposable elements in the response of fruit tree to climate change. As you know, transposable element and methylation are intimately linked, and it could be one way for fruit tree to cope with climate change. Why? Because actually when you have trees, they are grown for a lot of years, and they face climate every year, and it's always the same genotype. So methylation can play a role. I'm almost done. So it's a flexible way to play a role. And another thing is that why we have these trees, they have life history traits that they are perennial, and another, because they are perennial, we use grafting. We use grafting, and thanks to grafting, or I would say not thanks to grafting, because of that, we are starting to plant orchard with only one single clone in a lot of different fruit trees. And this has allowed the spread of disease like Erwinia, Xylella, Fusarium. So, as I show you in my presentation, domestication agriculture activity practices have consequences. And maybe actually we should go back to Vavilov thinking about whether we should better use diversity, favor gene flow between crop and wild and within crop, which could be the case beyond performance. But how to reconcile that and this view of agriculture to meet food demand and face climate change. So I try to put some idea here. One way would be just to change our use and practices, make orchard more diverse to limit the outbreak of disease, diversifying the cultures, that's one way. Another is what we have discussed today, is that using plant engineering, can we use that, like artificial and wide genetic diversity, but what are the consequences for the ecosystem? Especially in trees, you have a lot of gene flow among population, so we need to assess the risk and also develop some strategies to mitigate their impact. And so the take-home message is that we still have to do basic research on biodiversity to feed populations. And on that, I want to thank all the founder, also all my lab here, of course, of my group, and also all the New York University Abu Dhabi team, especially Stéphane, Pierre, Kirsten, Enas, Suzanne, Yves, Yann, Sandra, and Muriel for all the interaction and all of you. Thank you.